This is the lecture for translation. Translation is the process of creating a protein or a smaller version of a protein called a polypeptide using a messenger RNA created during transcription. Translation also has three phases: initiation, elongation, and termination. These are the same stages that are present during transcription. Okay, what components are needed for translation to occur properly? Well, first of all, you'll need the messenger RNA. That's the instructions used to determine which protein you're going to make and the order of the amino acids in the particular protein. You will need a transfer RNA. You will need an enzyme called aminoacyl tRNA synthetase, the ribosome, including both the small and the large subunit, the individual amino acids that are going to become a part of the protein, what are known as initiation factors, elongation factors, and a release factor. Now, some of these I didn't give any details with. We will be going over all of these as the lecture proceeds. All right, the messenger RNA. This is, again, containing the instructions to make that protein or polypeptide. Now, in this case, the messenger RNA is the instructions, including the sequence of which amino acids go in which place in the protein that you are creating. But how does that nucleotide sequence determine the amino acid sequence and which order they should be put into the protein? This is probably very familiar to you. It is known as the genetic code. You do not have to memorize this particular figure, but you should know how to use this table or a similar table given to you on an exam. So what this will do is it will take the amino acids that are given in the messenger RNA and translate them into a particular protein that is going to, a particular amino acid, an individual amino acid that will be added to the protein. Now, here it says codons up at the top. A codon is 3 nucleotides from the messenger RNA in a row. Okay. That's why you have, for the codons, you will have first position, second position, and third position. Those indicate the positions in the codon. Now, why do you need 3? Okay. You've probably gone through this before, but there are only 4 nucleotides in an RNA. Okay. You have the adenine, guanine, cytosine, and uracil. There's only 4. And those are represented by the letters here, U, C, A, and G. However, there are 20 different amino acids. So you can't say uracil represents a particular amino acid because there's only 4 nucleotides, 20 amino acids. But even if you took a sequence of 2 in a row, so for example, like UC would represent a particular amino acid, well, that wouldn't work either because that only gives 16 possibilities, and again, we have 20 amino acids. Therefore, it takes 3. So on here, for example, if the sequence from 5' to 3', for example, was, we'll just pick a particular example, C, G, A. That would be the sequence in the messenger RNA from 5' to 3' that would represent one codon. So to use this particular chart, C would be the first because that's at the 5' end. So you look for C at the 5' end under the first position. Then that would mean that it would be in this row. Then under the second position, you would look for, over here is second position, would be the G. So now you go to the G here, And that would mean this row. So that means where they intersect, that row and that column would be this particular box. So this row meeting up with this column would be that particular box. Now if you notice here, in this particular case, it doesn't matter what the last letter. Every single amino acid in that particular box is arginine. So the last letter will sometimes matter. This is the third position. Sometimes it won't. Now in the case I've just given you, it won't. But let's say we had, instead of CGA, we had, and I can put that over here, AGA. Well, if we look down here, the first letter is what changed. So here's A, then we go over to G again. Now, depending on what the third letter is, it does matter. If it was a U or a C, it would indicate serine. If it's an A or a G, it would represent arginine. So in this case, the A would represent arginine. This is how you read the table. So please make sure that you could do that on an exam. Okay. Now the other thing to notice on here is that most of the spaces and the inside part of the box here represent amino acids. But there are a few exceptions. One of them here, which is represented by AUG, which you should know, is what's called the start codon. This is going to be a codon that indicates starting of translation. You also have codons which are called stop codons, and those are over here. Okay. It's UAA, UAG, and UGA. Again, those you should know as well. So the start codons and the stop codons are, instead of representing a particular amino acid, represent an action to start or to stop. Now we know that a codon is 3 amino acids in a row, and they are going to indicate, that codon is going to indicate the particular amino acid that should be put in position at a particular spot. Okay. But it is important to keep in mind that what's known as a reading frame becomes very important. If we look at this particular case, first of all, when dealing with codons, you should always remember codon nucleotides, or a nucleotide that is used in a codon is never used again in a different codon. So all the codons have separate nucleotides. Okay. Now, if we look at this particular example of the reading frames, okay, remember a codon is 3 nucleotides in a row. So up here at the beginning, let's say we start with a particular frame of reference so that this begins our codon. So GCU becomes our first codon, then UGA, UAA, and so forth. If those represent the proper codons, and here notice there's a kind of isolated A out there, if you use those codons, you get a specific set of amino acids which would create a particular polypeptide. However, if now that first G is isolated by itself, and now the next letters, which are CUU, become a codon, that particular grouping will give rise to a very different polypeptide. That means that if the reading frame is not exact, if it's off by 1 nucleotide, it can mess up the entire polypeptide instructions. You may form a polypeptide, but obviously it's not going to be the same one. And if you're creating this one for a particular reason, creating this one in its place will not do. All right, just a reminder, messenger RNAs have to be created in the nucleus, so they must leave the nucleus through the nuclear pore complex to come out for translation to occur. Now this part here you covered earlier. There is a protein exchange. Remember when the RNA was exported through the nuclear pore complex, the cap binding complex was replaced by EIF4. That stands for eukaryotic initiation factor 4. Okay? And then the poly-A binding protein number 2 was replaced by poly-A binding protein number 1. So that's the protein exchange that occurred once the RNA has left the nucleus. We are going to be looking at those again shortly. Those particular molecules, the EIF4 and the poly-A binding protein 1 shortly. All right, what about transfer RNAs? What's their function and what do they do? Well, first of all, there are different types of transfer RNAs. One of them is called an initiator transfer RNA. Remember the start codon. The start codon, if you look at that particular genetic code chart again, there is also an abbreviation MET that is associated with the start codon. That stands for the amino acid methionine. The start codon, which is going to indicate the start of translation, also has an amino acid associated with it, which is methionine. Now, the initiator transfer RNA is going to be able to recognize that start codon, so it's going to recognize the starting point of translation. And also, very importantly, when it recognizes the start codon, it's going to get the correct reading frame. So that's very important here, the correct reading frame. The second thing it's going to do is it's going to have the first initial amino acid that's going to be associated with our protein, which is methionine. Now, during the elongation process, after initiation has started, additional TRAs for the different amino acids are going to be able to be needed to bring their own particular amino acid to the ribosome where translation is going to happen. This is a diagrammatic representation of a transfer RNA. If you'll notice, there is a stem portion called the acceptor stem. This is where the amino acid is going to bind. The purpose of a transfer RNA is to take a particular amino acid to the ribosome where the protein is being built. Okay. Now it also must be able to recognize that the amino acid it's carrying is the amino acid that should be put into that position in the growing protein. Now if that's a little bit confusing, it'll make more sense a little bit later. But there are different transfer RNAs for different amino acids. So the transfer RNA for methionine or for leucine is not going to be the same as for arginine. But if a particular transfer RNA is carrying lysine, it has to know that if it's going to be bringing the lysine into the ribosome, that it puts that lysine that it's carrying in the correct position in the growing chain. So the acceptor stem is going to be carrying the amino acid, and the amino acid I'm going to abbreviate as AA. The part of the transfer RNA that's going to help recognize that the amino acid it's carrying is the correct one is down here at what's called the anticodon loop. Okay. Now, the anticodon is appropriately going to be opposite or complementary to the codon, which is in the messenger RNA. So, if a codon has a particular amino acid sequence, the anticodon is going to be complementary. Now, there is a little bit of flexibility, but it's going to roughly be complementary to the codon strand. Now, what about the other loops, the D loop and the T-size CG loop? Okay. In this particular case, you should know there's other loops there, but I'm not going to hold you responsible for those really in detail. Those are just to allow the transfer RNA to be able to associate properly and move properly within the ribosome. All right, I also said you needed an enzyme which is called aminoacyl tRNA synthetase. Now the function of that particular enzyme is to take a transfer RNA with its loops and add the appropriate amino acid to that particular transfer RNA. Now in this diagrammatic representation, they've taken out the extra loops like the D loop just to make it easier to look at, but they're still there. Okay. So the aminoacyl tRNA synthetase is that big kind of goldish brown structure there, and that's going to be the enzyme. Notice it will have two binding sites, one of them for the separate amino acid, the separate binding site for the transfer RNA without its amino acid. Okay. Then it requires energy, and notice what it will do is it will associate the amino acid with the transfer RNA. Okay. That's its particular job, and it will be specific for that particular amino acid. Okay. Remember that each amino acid will have its transfer RNA that will be specific for it. All right, now in this particular case, all this is meant to represent is a little bit of the flexibility that you can see when you are dealing with the codon-anticodon interaction. On here, the upper figure is showing you what's called a wobble position, or the first position of the anticodon. Now here, this would correspond to the third position, or the wobble position, of the messenger RNA. Now what is a wobble position? Remember when we were looking back at the genetic code? For the messenger RNA, which was the codon, the third position sometimes mattered, sometimes didn't. Meaning, if it mattering or not is determined by whether, if you change that particular third nucleotide or the third part of the codon, whether or not the amino acid was changed. And again, sometimes it did and sometimes it didn't. So that's known as a wobble position because there's some variability. Well, on here, when you're looking at a transfer RNA, because the transfer RNA is complementary, the wobble position corresponds to position number 1 if it's read 5' to 3'. Okay. So if you look here, in red is the nucleotide that's found at that first position of the anticodon. And what you can see is in some cases there's a variability of the nucleotide that it will be able to recognize at that third position in the messenger RNA. Especially if there is a G, a U, or another nucleotide which is called inosine, which is the I, at that first position. Now the bottom figure just represents this kind of in a little bit different way. If you have these particular bases in the messenger RNA, in the wobble position or that third position of the messenger RNA, what you can see is that third position can be recognized by different nucleotides in the transfer RNA. Again, there's a little bit of flexibility there in the codon-anticodon interaction. Now, I'm not going to expect you to memorize this particular chart or figure. I won't ask you, for example, on here if it's G in the third wobble position of the messenger RNA, what possibilities are here? I will not ask you those types of questions. However, you should know in general that there is this flexibility between the messenger RNA codon and the anticodon in the transfer RNA. All right, ribosomes. Ribosomes are you can think of it as the location or kind of the machinery that's going to help carry out translation or protein synthesis. Now, we learned earlier that there was a large and a small subunit for ribosomes. However, they are distinguished by different representations based on whether if they're prokaryotic or eukaryotic. So for a prokaryotic ribosome, the large subunit is denoted as 50S. So sometimes in papers they will just say, oh, and there's a 50S. And they expect you to know that that's a prokaryotic and that it's the large subunit of the ribosome. The small subunit would be represented by 30S. For a eukaryotic ribosome, the large subunit is 60S and the small subunit is 40S. So if it helps you, try and remember prokaryotic or odd numbers, eukaryotic or even numbers. The other thing you should remember about ribosomes is that they are not only comprised of proteins. Ribosomes are comprised of proteins in combination with what are known as ribosomal RNA, or up here, rRNA. Now again on this figure, I'm not going to expect you to know the details of how many ribosomal RNAs are in each subunit. That isn't really important for this class, but what you do need to remember is yes, there are ribosomal RNA subunits associated with ribosomes, along with proteins, and why they're there. Now why they're there, remember what's going to be binding to and associating with ribosomes. The messenger RNA and transfer RNA, both of those are RNA. If the ribosome was just made out of proteins, it would make the interaction between the ribosome and the transfer RNA and the messenger RNA much more difficult. By having the ribosomal RNA associated with the ribosome, along with proteins, it makes that interaction with messenger RNA and transfer RNA much more efficient and stable. All right, now we can get into the process of how translation occurs. Let's start with initiation of translation, and remember, initiation is often the most complex portion. All right, so the subunits for the ribosome. Here, obviously, since they're even numbers, should remember this represents then a eukaryotic organism. The ribosome goes back and forth between a state where the subunits are associated with each other and a condition where they're disassociated or separate from each other. Now we are going to start with them separated from each other. So we have a separate 60S subunit and a separate 40S subunit. Now if we were dealing with a prokaryotic organisms, remember it would be 50S and 30S. All right, the 40S subunit, which is the small subunit, okay, and you should remember that, okay. The small subunit is associated with its own initiation factor, here represented by the little green number 3. So it's an initiation factor 3. Now here what you can kind of think of it is, it will help to ensure that what's going to happen next happens in the correct location. So what it's doing is it's helping to set up the next steps following. Okay. Then there's an initiation factor 1A, which will join the small subunit. Now, to help make a sense of this, hopefully you remember from previous classes that the subunits, both the large and the small subunit, have positions or slots inside of them. It doesn't matter what you call them, positions or slots, but just remember they have these positions or slots, okay? And that's where transfer RNAs or other like initiation factors are going to sit. And it goes E, P, and A. Okay. So at the end of this part, you're going to have one initiation factor sitting in one slot and another initiation factor sitting in another slot. And the middle slot, which is the P slot, is going to be the only one open. Now, that's going to be important because that's what's going to allow a initiator transfer RNA, and that's what's kind of represented by that little I there. And remember, the initiator transfer RNA has the first amino acid that's going to be in this particular protein, which is methionine, the MET, associated with it. Now, it also has a initiation factor associated with it, which is called EIF2. That EIF2 stands for eukaryotic initiation factor number 2, and it's going to be associated with the initiator transfer RNA. Notice this initiation factor 2 is a molecule that is bound to GTP. That will be important later. GTP, remember, is going to be a higher energy form, so this is a higher energy molecule at the moment. That initiator transfer RNA attached to methionine and that initiator transfer RNA bound to initiation factor 2, and that's associated with the GTP, so that whole entire complex is located in the position number P of the ribosome. What happens next? Okay, so the upper figure up here is from the previous slide. Now the messenger RNA is going to be coming in. Here is that eukaryotic initiation factor 4. That's the one you learned from before that replaced the original cap binding complex. Okay. Now, it's still the initiation factor 4 is still binding to the 5' end. So here it's a little misleading, but it's still binding to the 5' cap region. So it is just a different type of a cap binding complex. Okay. Now here, that initiation factor 4, one of its jobs and why it's important is it's going to allow the ribosomal subunit to find the 5' end of the messenger RNA. Remember that in translation, the messenger RNA is going to be read from 5' to 3'. So the messenger RNA needs to start at the right end of the ribosome. It needs to find the ribosome at the correct end. You don't want to have the 3' end of the messenger RNA bind to the ribosome. So that eukaryotic initiation factor 4 is going to help the ribosome complex, which is the small subunit, find the correct end of the messenger RNA, which is the 5' end. Once it's done that, it's now correctly found the 5' end, but now there's a little bit of a problem. We're not ready to kind of delve into translation yet. Okay. The initiator transfer RNA is not at the correct location. It knows it's not at the correct location because its anticodon does not meet up with the codon. Remember the start codon, which is AUG. On the figure up here, it's way over here. Our initiator transfer RNA is over here. They are not met up. It's important for it to actually find the correct AUG. That's how remember, it gets the correct reading frame. So this initiator transfer RNA must find its way to there. Okay. So what will happen now is, with the help of an energy source, it's going to start moving down the messenger RNA. And it will keep moving until you have the AUG find its match at the anticodon, which is on the transfer RNA. So that's the anticodon for the AUG. Only when that happens, and again, only when this match occurs, then you have the eukaryotic initiation factor 2 hydrolyze the GTP that was on there to GDP and the phosphate, and it will leave. So it's showing on here all of this leaves from this initiation complex, which was before. Notice what else leaves. The initiation factor 1A and 3 that were also positioned within the ribosomal subunit, they leave as well, along with much of the initiation factor 4 complex that was helping to position the messenger RNA correctly, because it's no longer needed. Now here I say much of because it seems that part of the initiation factor 4 complex actually will stay on the cap part of the messenger RNA. All right, so now what we have is the initiator transfer RNA. It's lost its initiation factor 2, which is an association factor. It helps things proceed correctly. But you've lost the initiation factor 2, which was needed to help find the start codon. But now that the start codon is found, that's lost. You've lost the initiation factors 1A and 3 that were part of the ribosomal complex. And so now you're ready for kind of the last steps in initiation. Here, what we have to do to complete initiation is to join the large subunit of the ribosome to the small subunit to complete the initiation process. Okay, so here's the 60S subunit with its own initiation factor, which is number 6. It will help position the large subunit correctly with the small subunit so that the E slot matches up with the E slot correctly on the small subunit, and the P slot matches up with the P slot correctly, and so forth. That's what the initiation factor 6 does. Okay. And once the two subunits join together, you now have a complete ribosome with the initiator transfer RNA inside the P slot, which is represented there. and the messenger RNA positioned correctly within the ribosome. Okay. At this point, the initiation of translation is completed. Okay, now we can go on to elongation. We are now starting off with where we left off before. This here is the initiator transfer RNA bound to the initial amino acid, which is methionine, which is right there. Notice it's located in the P slot. Here there's a little bit more detail with the ribosome showing the E, the P, and the A slot. Now we are ready to go on and add the next amino acid to this growing peptide chain that we're going to start here. Notice off to the side here we have a series of transfer RNAs, each bound to an amino acid. Now here the amino acids are represented in a general term. The numbers 2, 3, and 4 represent different amino acids. Now here they also represent, because they're sequential numbers, the order in which they're supposed to join the growing peptide chain. So here this one should be the second one that joins, this one the third, this one the fourth. Now what we're going to do is we're going to start growing the peptide chain. So we're going to have the next amino acid, which should be number 2 here, join the ribosome so that the second amino acid could be joined to the first one, which is methionine from before. However, it becomes a question as the second one is meant to join the ribosome. However, all of these transfer RNAs with their amino acids are just floating around in the cytoplasm. So in theory, it is just as likely that number 4, which is wrong, could join the ribosome instead of number 2. How do we know that 2 will get added correctly rather than 4 incorrectly? Here we have a separate molecule, which is a protein which is called EF1, and you can ignore the alpha. EF1 stands for elongation factor 1. Elongation factor 1 is associated with the GTP, so that should tell you it's a high energy form. And it's associated with every transfer RNA. Think of it as like a weight. So let's just say, for example, over here, our transfer RNA carrying amino acid number 4 tries to join the ribosome, but it's not its correct turn. It's not correct. How do we know that it's not the correct one? Well, remember the instructions on which amino acid should be added next is going to be in the messenger RNA here, and it's going to be at the A slot. Okay. For that reason, when you have a transfer RNA joining into the A slot, the anticodon that is on the transfer RNA should match the codon, again complementarily, in a complementary way, to the codon that's in the A slot. Okay. If they don't match, it's not a stable bond. Remember, if they match, you should be able to form hydrogen bonds, and so there's kind of a stability there. If the codon and the anticodon do not match, there is no stability. And that elongation factor 1 that acts kind of like a weight will pull the wrong transfer RNA out of the ribosome. And so then what will happen is it will leave. Okay, it won't be able to stay in the ribosome. However, if it is the correct transfer RNA, what will happen is it does match the codon. And so then you get stability. And what that will do is it will allow the next step to proceed. Okay, so on here we now have the next correct amino acid joining the A slot. So again here, the codon and the anticodon match. Anticodon are going to match. What that's going to do is it's going to create stability. That will trigger hydrolysis of the GTP into GDP and phosphate, and elongation factor 1 will leave. So think of it, the job of elongation factor 1 is to act almost like a weight to pull away any transfer RNA that's incorrectly in the A slot of the ribosome. If the correct transfer RNA adds, that's going to cause the GTP to hydrolyze to GDP and phosphate, and the elongation factor will leave because it's no longer needed. Now at this point, you're going to have an association of the amino acids number 1 and number 2 from the two different transfer RNAs, one in the P slot and one in the A slot. This is going to assist in the peptide bond formation between amino acid number 1 and amino acid number 2. Now notice at this case where amino acid number 1 ends up, okay? It leaves the transfer RNA, okay, and joins to the amino acid number 2. So now both amino acids are associated with the one transfer RNA that's in slot A. That's very important, that the amino acid that's in slot number P leaves the transfer RNA, disassociates from the transfer RNA in slot P, and joins with the amino acid in slot on the transfer RNA in slot A. Now what will happen is an elongation factor number 2 associates with the transfer RNA in the A slot. So here's elongation factor 2. Notice it also has a GTP associated with it, so it's a higher energy molecule. And it will come and associate with the transfer RNA in slot A. Now, part of its job is to make sure that the transfer RNAs move to the next position in the ribosome. So here what happens is everything slides down, including the messenger RNA. So what was in the P slot moves to the E slot, what was in the A slot moves to the P slot. And notice the messenger RNA moves with it. Okay. Now here, elongation factor 2 seems to be associated primarily with this migration of the transfer RNAs. And that seems to be the case. But there is also some indication that elongation factor 2 might also play a role in the previous step, which was the formation of that peptide bond. Okay, now we have the transfer RNA that is now empty, that was in the P slot, in the E slot. We now have the transfer RNA bound to both amino acids 1 and 2 in the P slot, and now we have an empty A slot. Okay. Now the last part here, and this will continue in the next slides, is that once you have a transfer RNA in the E slot, it will actually leave. So the transfer RNA will then actually leave. And you can think of E standing for exit, where the transfer RNA leaves. We'll now go over what happens with termination. Okay, here we're picking up the process much further along. If you notice, we have added many amino acids to our growing strand. All of this occurred by the process of elongation. Now, previously when we went over elongation, we just added one amino acid to another one. However, this process will just keep repeating over and over again during elongation. So once one amino acid has been added, you can start the process again and add another one until you get a long chain like this one. Each part of elongation will proceed just like the step before. But at some point, elongation is going to have to stop or end. And so then we get to the point where we want to stop translation. That is indicated by the presence of the stop codon in the slot A. So you can see UAA there representing a stop codon. Now once we have a stop codon there, we now have an indication that this is the end of translation. There is no real transfer RNA that can recognize a stop codon. However, what there is what's known as a release factor. And that's indicated right here as ERF1 for eukaryotic release factor 1. It is associated with its own release factor assistant, okay, which is known as release factor 3 associated with it. Release factor 3 is a higher energy molecule, again associated with GTP. Now here, once the release factor recognizes the anticodon, which is present in the messenger RNA down there, once that is correctly recognized, the release factor 3 hydrolyzes the GTP to GDP, and then simply what happens is at this point is the whole complex becomes unstable. There is no amino acid on the release factor for the peptide to join to. So once conformation changes happen because of the hydrolysis here of GTP to GDP and phosphate, the whole complex becomes unstable. And notice everything just kind of falls apart. The transfer RNAs leave the ribosomes. The peptide, which you have just formed, or the protein if it's longer, can leave and go do whatever function it's supposed to do. It's not really showing it on here, but the messenger RNA is also there. Okay. And what will happen is the peptide will go do its function, the transfer RNAs are recycled, the ribosomes are recycled. The messenger RNA, depending on the signals in the cell, will either be used to make more protein, so it will join with another ribosome and make more protein, or it will be degraded. This slide is showing an important factor in translation. It's often thought of that there's one ribosome on a particular messenger RNA, but that's usually not the case. You can see on here the messenger RNA represented by the red line with the 5' end over here and the poly A tail here, can often be associated with multiple ribosomes at one time, meaning that you can have a complex with the messenger RNA and many ribosomes all translating the same messenger RNA at the same time. This makes it much more efficient because you don't need to create proteins individually. You can take one messenger RNA and essentially be creating multiple proteins at the same time from that messenger RNA. This can sometimes be very efficient because sometimes you'll need a larger quantity of the proteins, and this way you can make it much faster than if you had to individually make the proteins from the messenger RNA, so that the messenger RNA would make one protein and then you may have to wait till it finishes and then make another one. That may be much slower, and if you need a larger number of the proteins, it could take too long. Okay, this is the end of the translation. If you have any questions, please talk to me about this in class or during my office hours.